Today we are going to see about microbial assay of antibiotics and amino acid. Introduction. A microbial assay is a biological method used to determine the potency or concentration of antibiotics and amino acid by measuring their effect on the growth of specific microorganism. These assays are based on the principle that microbial growth is directly proportional to the amount of biological active substance present. Microbial assays are widely used because they measure biological activity, not just chemical concentration. Microbial assay of antibiotics. Principle. Antibiotics inhibit the growth of susceptible microorganisms. When an antibiotic diffuses into a nutrient medium seeded with a test organism, it produces zone of inhibition or reduced microbial growth. The size of the inhibition zone or degree of growth reduction is proportional to the antibiotic concentration. Common test organism for antibiotics. Different antibiotics require specific test organism. Antibiotics test organism: penicillin, Staphylococcus aureus, streptomycin, Bacillus subtilis, tetracycline, Bacillus cereus, chloramphenicol, E. coli. Method of antibiotic assay. Agar diffusion method. This is the most commonly used method. Cylindrical plate method. Sterile cylindrical are placed on the agar plate seeded with test bacteria. Standard and test antibiotic solution are added to the cylinders. Plates are incubated at 37 degrees Celsius for 18 to 24 hours. Zone of incubation are measured. Potency is calculated by comparing test and standardized zone. Cup plate method. Wells are cut in the agar instead of using cylinders. Antibiotic solution are added to the well. Zone diameter indicates antibiotic activity. Turbidimetric method. Liquid nutrient medium is inoculated with test organism. Antibiotic inhibit microbial growth. Growth is measured using a spectrophotometer. Low turbidity indicates higher antibiotic concentration. Advantage of antibiotic assay. Measures biological effectiveness. Simple and economical. Suitable for routine quality control. Limitation. Time consuming. Sensitive to environmental conditions like pH, temperature. Less precise than chemical method. Microbial assay of amino acid. Principle. Certain microorganisms cannot synthesize specific amino acid and require them in their growth medium. The growth of this organism is directly proportional to the concentration of the amino acid supplied. Common test organism for amino acid. Amino acid test organism. Lysine, leuconostoc mesenteroids. Methionine, Streptococcus faecilae. Arginine, Lactobacillus arabinosus. Tryptophan, Lactobacillus plantarum. Assay method. Turbidimetric method. Procedure. Prepare a basal medium lacking the amino acid to be tested. Inoculate with a specific axonotropic microorganism. Add known concentration of standard amino acid and test sample. Incubate at 37 degrees Celsius for 18 to 24 hours. Measure turbidity using a spectrophotometer. Plot a standard growth curve. Determine unknown concentration from the curve. Factor affecting amino acid assay. pH of the medium, incubation temperature, purity of culture, presence of interfering substance. Advantage of amino acid. Highly sensitive, measure biological active amino acid, useful in food and pharmaceutical industry. Limitation. Slow process, requires strict aseptic condition, not suitable for mixed amino acid sample. Application of microbial assays. Quality control of antibiotics, estimation of vitamin and amino acid, pharmaceutical standardization, food and feed analysis, clinical and research laboratories. Thank you.